Welcome to a deep dive into the world of viruses. You've brought excerpts from the Color Atlas of Medical Virology, so I'm guessing you're ready to explore these microscopic wonders.
Viruses are definitely more than just microscopic. They're masters of adaptation, constantly evolving, and their story is really intertwined with our own.
Exactly. It's fascinating how something so small has had such a huge impact. So before we get into the details, what makes viruses so unique?
Well, it's even debated if they're alive. They're not like bacteria, which are single-celled organisms. Viruses are much simpler.
How so?
They're basically just packets of genetic material, either DNA or RNA, inside a protein coat. They vary a lot in shape and size, but that basic structure is always there.
Like the bare essentials of genetic information almost.
Yeah, exactly. And that's what makes them such pros at getting into our cells. They're what we call obligate intracellular parasites. They can't reproduce on their own. They need to hijack a host cell's machinery to make copies of themselves.
Okay, that's both clever and unnerving. So how does a virus go from just existing outside a cell to, like, taking it over?
It's all about their ability to recognize and bind to specific cells. It's like viral proteins are keys, each one fitting a particular lock on a cell's surface.
So they find their target, what happens next?
Their simple structure is their strength here. Once that viral protein key finds its lock on the cell, it's like a chain reaction. The virus can slip inside, either by merging with the cell membrane or finding a back door.
And once they're in, the cell's doomed, right? It just becomes a virus factory.
Pretty much. The virus releases its genetic material, and it's like a set of instructions, hijacking the cell to make more of the virus.
The Color Atlas mentions viral proteins like hemagglutinin in influenza. What do those do?
Those are crucial for the takeover. Hemagglutinin, for example, is how the flu latches onto cells in your respiratory tract, like a grappling hook.
So each viral protein has a specific job in this whole infection process. But if viruses are so good at this, how does our body ever stand a chance?
That's where our immune system comes in, a remarkable defense system that's constantly evolving to fight these invaders.
So it's a constant back and forth, viruses attacking and our bodies defending.
Absolutely, a constant battle that shaped both viruses and ourselves over millions of years.
This is already mind-blowing. But before we get to the immune response, let's go back to something you said earlier. Viruses can have DNA or RNA as their genetic material. What difference does that make?
That's a really important point. The type of genetic material a virus has impacts how it replicates, how it evolves, and how it interacts with us.
So we need to decode those differences to really understand viruses. I'm ready.
Okay, so DNA. We know that's like the blueprint of life, right? Stable, passed down through generations. RNA is different. It's more like a temporary message, a working copy of those DNA instructions that helps build proteins.
And viruses can use either one. Wow. But what's the big difference between, say, a DNA virus and an RNA virus? How do they act differently?
Think of DNA viruses like the planners, the strategists. They're usually more stable, doing their thing inside the cell's nucleus, sometimes even weaving themselves into the host's DNA.
And that's not always a bad thing, right? I mean, our DNA actually contains bits of ancient viruses, doesn't it?
You're right. Some viral DNA is now a part of us. But with DNA viruses like HPV, which is linked to some cancers, or herpes viruses, well, that integration can be a problem long term.
So they can lie dormant for years. It's kind of creepy, but also fascinating.
And then there are RNA viruses. Those are the ones that change quickly, the masters of adaptation. RNA is naturally less stable than DNA, so these viruses, they mutate a lot.
That explains why it's so hard to make a flu vaccine that lasts. There's always a new strain.
Exactly. It's like trying to hit a moving target. You know, the Color Atlas talks about this with antigenic drift and antigenic shift.
Those sound kind of intense. Can you explain?
Okay, picture the flu virus as a chameleon, always changing to blend in. Antigenic drift is like a subtle change in its camouflage, small changes in its proteins that happen over time. Our immune system might still kind of recognize it, but it's not as effective.
So it's like the virus is constantly fine-tuning, staying a step ahead, and antigenic shift. That's the big one.
That's a total makeover. It's when two different flu viruses infect the same cell and they swap genetic material. Creates a totally new hybrid virus with new proteins.
So our immune system is totally caught off guard. That's why those pandemic strains are so scary.
It's like the virus learned a whole new language. But, you know, as challenging as this is, it shows how incredible viruses are at survival.
You mentioned how some DNA viruses can integrate into our DNA. That's a pretty powerful adaptation, too.
Absolutely. That ability to become part of our DNA is amazing. But it can backfire. Sometimes it messes with normal cell growth and can even lead to cancer.
Wow, viruses can really impact our biology. The Color Atlas also mentioned a possible link to other diseases like diabetes. How does that work?
That's still being researched, but there's more and more evidence that some viruses might trigger our immune system to attack our own insulin-producing cells. It shows just how complex the relationship is between viruses and our immune systems.
It seems like the more we learn, the more complex it gets, but no matter what, our bodies need to fight back. So let's talk about how our immune system actually tackles these tiny invaders. It's amazing how our bodies have this whole defense system in place. But how do we even know what we're dealing with? Viruses are invisible. How do you diagnose a viral infection?
Well, we can't just look at them and know, that's for sure. The Color Atlas talks about different methods, from culturing viruses to actually finding their genetic material. It's pretty ingenious.
Viral detective work, what's the most common way to diagnose?
We often start with something called virus isolation in cell culture. Sounds complicated, but it's pretty straightforward. We take a sample from the patient, could be blood or a swab, and we add it to a dish of cells that we grow in the lab. If there's a virus, it infects the cells, and we can study it.
So it's like giving the virus a place to multiply so you can see what's going on. Then what?
Then we have ways to identify it. One way is immunofluorescence. We use antibodies that are designed to attach to specific viral proteins. These antibodies have fluorescent molecules on them, so when you look at the infected cells under a microscope, the viral proteins light up.
Like a microscopic spotlight. I love
Yeah,
it. What other methods are there?
Antigen detection tests becoming more popular. Those tests don't need to grow the virus in a culture. They can detect specific viral proteins, called antigens, right in the patient's sample. It's kind of like those home pregnancy tests, but for viruses.
Wow, much faster. And then there are PCR tests, of course. Seems like everyone knows about those now.
Right. PCR, which stands for polymerase chain reaction, can make copies of even tiny amounts of viral DNA or RNA. So even if there's not much of the virus, we can find it. Super useful for viruses that are hard to grow in the lab.
It's incredible how far we've come in understanding viruses. But how do we actually fight them? It's not like bacteria. We can't just use antibiotics.
Exactly. Viruses are much trickier because they live inside our own cells. It's hard to kill the virus without hurting the cell itself.
It's like trying to remove a splinter that's inside a delicate bubble. What can we do?
We're not totally helpless. We have antiviral drugs now. They're designed to attack specific parts of the virus, like certain enzymes or proteins it needs to replicate.
Can you give me some examples?
Sure. Acyclovir, for example, is used for herpesvirus infections. And oseltamivir, or Tamiflu, as it's better known, can help shorten the flu.
So it's about finding the virus's weak spots. But prevention is key, right? That's where vaccines are so important.
Absolutely. Vaccines are one of the best tools we have. They work by using our immune system's ability to learn and remember.
So it's like giving our immune system a sneak peek of the virus so it's ready for a real attack later. The Color Atlas mentions different kinds of vaccines. What's the difference?
Live attenuated vaccines use a weak version of the virus. This weakened version can still replicate, but it's very unlikely to make you sick. But it gives your immune system a good workout.
Like a training exercise for your immune system. What about inactivated vaccines?
Inactivated vaccines use a dead version of the virus. It can't replicate anymore, but it still has those important viral proteins that our immune system can recognize. The flu shot is a good example.
So with inactivated vaccines, it's like showing your immune system a picture of the bad guy and saying, watch out for this one. But we've talked about how much viruses change, especially those RNA viruses. Does that make it harder to make vaccines?
It definitely can. That whole antigenic drift and shift thing makes it tricky to create vaccines that last a long time, especially for some viruses.
Like trying to hit a moving target.
Right. Exactly. It's a constant challenge. That's why we often need booster shots or new vaccines every year, like for the flu. Scientists are always on the lookout for new strains and tweaking vaccines to keep up.
It's a never-ending battle. Well, this has been an amazing deep dive. We've learned about how viruses infect us, how our immune system fights back, and what we're doing to stay one step ahead. It's a lot to take in.
It really is. And it's just the tip of the iceberg, really. There's still so much we don't know about viruses, but we're learning more all the time. New viruses, new treatments, and new challenges are always emerging.
It makes you realize how connected we are to this tiny world, even if we can't see it. And knowledge is power, right? The more we understand, the better prepared we are. Thank you so much for joining us on this incredible journey into the world of viruses. Until next time, stay curious.